Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Microtubule. 52 languages. Microtubule and tubulin metrics. 1. Microtubules are one of the cytoskeletal filament systems in eukaryotic cells. The microtubule. Microtubules are polymers of tubulin that form part of the cytoskeleton and provide structure and shape to eukaryotic cells. Microtubules can be as long as 50 micrometers, as wide as 23 to 27 nm, 2, and have an inner diameter between 11 and 15 nm. 3. They are formed by the polymerization of a dimer of two globular proteins, alpha and beta tubulin, into protofilaments that can then associate laterally to form a hollow tube, the microtubule. 4. The most common form of a microtubule consists of 13 protofilaments in the tubular arrangement. Microtubules play an important role in a number of cellular processes. They are involved in maintaining the structure of the cell and, together with microfilaments and intermediate filaments, they form the cytoskeleton. They also make up the internal structure of cilia and flagella. They provide platforms for intracellular transport and are involved in a variety of cellular processes, including the movement of secretory vesicles, organelles, and intracellular macromolecular assemblies. 5. They are also involved in cell division, by mitosis and meiosis, and are the main constituents of mitotic spindles, which are used to pull eukaryotic chromosomes apart. Microtubules are nucleated and organized by microtubule organizing centers, such as the centrosome found in the center. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Cytoskeleton is involved in the transport of material within cells, carried out by motor proteins that move on the surface of the microtubule. Of many animal cells or the basal bodies of cilia and flagella, or the spindle pole bodies found in most fungi. There are many proteins that bind to microtubules, including the motor proteins dynein and kinsin, microtubule severing proteins like catenin, and other proteins important for regulating microtubule dynamics. 6. Recently an actin-like protein has been found in the gram-positive bacterium Bacillus thuringiensis, which forms a microtubule-like structure called a nanotubule, involved in plasmid segregation. 7. Other bacterial microtubules have a ring of 5 protofilaments. History, edit. Tubulin and microtubule-mediated processes, like cell locomotion, were seen by early microscopists, like Leeuwenhoek, 1677. However, the fibrous nature of flagella and other structures were discovered 2 centuries later, with improved light microscopes, and confirmed in the 20th century with the electron microscope and biochemical studies. 8. In vitro assays for microtubule motor proteins such as dynein and kinsin are researched by fluorescently tagging a microtubule and fixing either the microtubule or motor proteins to a microscope slide, then visualizing the slide with video-enhanced microscopy to record the travel of the motor proteins. This allows the movement of the motor proteins along the microtubule or the Microtubule moving across the motor proteins. 9. Consequently, some microtubule processes can be determined by chymograph. 10. Structure, edit. In eukaryotes, microtubules are long, hollow cylinders made up of polymerized alpha and tubulin dimers. 12. The inner space of the hollow microtubule cylinders is referred to as the lumen. The alpha and tubulin subunits are 50% identical at the amino acid level, and both have a molecular weight of approximately 50 K. The 13, 14. These alpha/tubulin dimers polymerize end-to-end into linear protofilaments that associate laterally to form a single microtubule, which can then be extended by the addition of more alpha/tubulin dimers. Typically, microtubules are formed by the parallel association of 13 protofilaments, although microtubules composed of fewer or more protofilaments have been observed in various species. 15, as well as in vitro. 16. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Cartoon representation of the structure of alpha, yellow, / beta, red. Tubulin heterodimer, GTP and GDP. 11. Microtubules have a distinct polarity that is critical for their biological function. Tubulin polymerizes end-to-end, with the beta subunits of one tubulin dimer contacting the subunits of the next dimer. Therefore, in a protofilament, one end will have the subunits exposed while the other end will have the subunits exposed. These ends are designated the and plus ends, respectively. The protofilaments bundle parallel to one another with the same polarity, so, in a microtubule, there is one end, the plus end, with only subunits exposed, while the other end, the end, has only subunits exposed. While microtubule elongation can occur at both the plus and ends, it is significantly more rapid at the plus end. 17. The lateral association of the protofilaments generates a pseudo-helical structure, with one turn of the helix containing 13 tubulin dimers, each from a different protofilament. In the most common 13. 3 architecture, the 13th tubulin dimer interacts with the next tubulin dimer with a vertical offset of 3 tubulin monomers due to the helicity of the turn. There are other alternative architectures, such as 11-3, 12-3, 14-3, 15-4, or 16-4, that have been detected at a much lower occurrence. 18. Microtubules can also morph into other forms such as helical filaments, which are observed in protist organisms like foraminifera. 19. There are two distinct types of interactions that can occur between the subunits of lateral protofilaments within the microtubule called the A-type and B-type lattices. In the A-type lattice, the lateral associations of protofilaments occur between adjacent alpha and tubulin subunits, i.e. an tubulin subunit from one protofilament interacts with a tubulin subunit from an adjacent protofilament. In the B-type lattice, the alpha and tubulin subunits from one protofilament interact with the alpha and tubulin subunits from an adjacent protofilament, respectively. Experimental studies have shown that the B-type lattice is the primary arrangement within microtubules. However, in most microtubules there is a seam in which tubulin subunits interact alpha, beta. 20. The sequence and exact composition of molecules during microtubule formation can thus be summarized as follows. A tubulin connects in the context of a non-existent covalent bond with an tubulin, which in connected form are a heterodimer, since they consist of two different polypeptids, tubulin and tubulin. So after the heterodimers are formed, they join together to form long chains that rise figuratively in one direction, for example upwards. These heterodimers, which are connected in a certain direction, form protofilaments. These long chains, protofilaments, now gradually accumulate next to each other so that a tube-like structure is formed, which has a lumen typical of a tube. Accordingly, mostly 13 protofilaments form the outer wall of the microtubules. The heterodimers consist of a positive and negative end, with alpha tubulin forming the negative end, and beta tubulin the positive end. Due to the fact that the heterodimers are stacked on top of each other, there is always a negative and positive end. Microtubules grow by an addition of heterodimers at the plus end. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Components of the eukaryotic cytoskeleton. Actin filaments are. Some species of Prosthecobacter also contain microtubules. The structure of these bacterial microtubules is similar to that of eukaryotic microtubules, consisting of a hollow tube of protofilaments assembled from heterodimers of bacterial tubulin A, Bdub-A, and bacterial tubulin B, Bdub-B. Both Bdub-A and Bdub-B share features of both alpha and tubulin. Unlike eukaryotic microtubules, bacterial microtubules do not require chaperones to fold. 21. In contrast to the 13 protofilaments of eukaryotic microtubules, bacterial microtubules comprise only 5. 22. Intracellular organization, edit. Microtubules are part of the cytoskeleton, a structural network within the cell's cytoplasm. The roles of the microtubule cytoskeleton include mechanical support, organization of the cytoplasm, transport, motility and chromosome segregation. In developing neurons, microtubules are known as neurotubules. 23, and they can modulate the dynamics of actin, another component of the cytoskeleton. 24. A microtubule is capable of growing and shrinking in order to generate force, and there are motor proteins that allow organelles and other cellular components to be carried along a microtubule. This combination of roles makes microtubules important for organizing and moving intracellular constituents. The organization of microtubules in the cell is cell type specific. In epithelia, the minus ends of the microtubule polymer are anchored near the site of cell-cell contact and organized along the apical basal axis. After nucleation, the minus ends are released and then re-anchored in the periphery by factors such as 9 and PLK7. 25. In this manner, they can facilitate the transport of proteins, vesicles and organelles along the apical basal axis of the cell. In fibroblasts and other mesenchymal cell types, microtubules are anchored at the centrosome and radiate with their plus ends outwards towards the cell periphery, as shown in the first figure. In these cells, the microtubules play important roles in cell migration. Moreover, the polarity of microtubules is acted upon bimotor proteins, which organize many components of the cell, including the endoplasmic reticulum and the Golgi apparatus. Microtubule polymerization, edit. Nucleation, edit. Main article, Microtubule nucleation. Nucleation is the event that initiates the formation of microtubules from the tubulin dimer. Microtubules are typically nucleated and organized by organelles called microtubule organizing centers, MTOCs. Contained within them is another type of tubulin, which is distinct from the alpha and subunits of the microtubules themselves. The tubulin. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Shown in red, microtubules are in green, and the nuclei are in blue. The cytoskeleton provides the cell with an inner framework and enables it to move and change shape. Combines with several other associated proteins to form a lock washer-like structure known as the tubulin ring complex, gamma 2RC. This complex acts as a template for alpha-tubulin dimers. To begin polymerization, it acts as a cap of the end while microtubule growth continues away from them in the plus direction. 26. The centrosome is the primary MDOC of most cell types. However, microtubules can be nucleated from other sites as well. For example, cilia and flagella have MTOCs at their base termed basal bodies. In addition, work from the Cavrina group at Vanderbilt, as well as others, suggests that the Golgi apparatus can serve as an important platform for the nucleation of microtubules. 27. Because nucleation from the centrosome is inherently symmetrical, Golgi-associated microtubule nucleation may allow the cell to establish asymmetry in the microtubule network. In recent studies, the Vail group at UCSF identified the protein complex augmin as a critical factor for centrosome-dependent spindle-based microtubule generation. That has been shown to interact with 2RC and increase microtubule density around the mitotic spindle origin. 28. Some cell types, such as plant cells, do not contain well-defined MTOCs. In these cells, microtubules are nucleated from discrete sites in the cytoplasm. Other cell types, such as trypanosomatid parasites, have a MTOC but it is permanently found at the base of a flagellum. Here, nucleation of microtubules for structural roles and for generation of the mitotic spindle is not from a canonical centriole-like MTOC. Polymerization, edit. Following the initial nucleation event, tubulin monomers must be added to the growing polymer. The process of adding or removing monomers depends on the concentration of tubulin dimers in solution in relation to the critical concentration, which is the steady-state concentration of dimers at which there is no longer any net assembly or disassembly at the end of the microtubule. If the dimer concentration is greater than the critical concentration, the microtubule will polymerize and grow. If the concentration is less than the critical concentration, the length of the microtubule will decrease. 29. Microtubule dynamics, edit. Dynamic instability, edit. Dynamic instability refers to the coexistence of assembly and disassembly at the ends of a microtubule. The microtubule can dynamically switch between growing and shrinking phases in this region. 30. tubulin dimers can bind 2 molecules of GTP, one of which can be hydrolyzed. Subsequent to assembly. During polymerization, the tubulin dimers are in the GTP-bound state. 12. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Animation of the microtubule. Dynamic instability. Tubulin dimers. Bound to GTP, red, bind to the growing end of a microtubule and subsequently hydrolyze GTP into GDP, blue. The GTP bound to tubulin is stable and it plays a structural function in this bound state. However, the GTP bound to beta tubulin may be hydrolyzed to GDP shortly after assembly. The assembly properties of GDP tubulin are different from those of GTP tubulin, as GDP tubulin is more prone to depolymerization. 31. A GDP-bound tubulin subunit at the tip of a microtubule will tend to fall off, although a GDP-bound tubulin in the middle of a microtubule cannot spontaneously pop out of the polymer, since tubulin adds onto the end of the microtubule. In the GTP-bound state, a cap of GTP-bound tubulin is proposed to exist at the tip of the microtubule, protecting it from disassembly. When hydrolysis catches up to the tip of the microtubule, it begins a rapid depolymerization and shrinkage. This switch from growth to shrinking is called a catastrophe. GTP-bound tubulin can begin adding to the tip of the microtubule again, providing a new cap and protecting the microtubule from shrinking. This is referred to as rescue. 32. Search and capture model, edit. In 1986, Mark Kirschner and Tim Mitchison proposed that microtubules use their dynamic properties of growth and shrinkage at their plus ends to probe the three-dimensional space of the cell. Plus ends that encounter kinetic arrays or sites of polarity become captured and no longer display growth or shrinkage. In contrast to normal dynamic microtubules, which have a half-life of 5–10 minutes, the captured microtubules can last for hours. This idea is commonly known as the search and capture model. 33. Indeed, work since then has largely validated this idea. At the kinetic, a variety of complexes have been shown to capture microtubule plus ends. 34. Moreover, a plus end capping activity for interface microtubules has also been described. 35. This later activity is mediated by formins. 36. The adenomatous polyposis coli protein, and EB1. 37. A protein that tracks along the growing plus ends of microtubules. Regulation of microtubule dynamics, edit. Post-translational modifications, edit. Although most microtubules have a half-life of 5–10 minutes, certain microtubules can remain stable for hours. 35. These stabilized microtubules accumulate post-translational modifications on their tubulin subunits by the action of microtubule-bound enzymes. 38, 39. However, once the microtubule depolymerizes, most of these modifications are rapidly reversed by soluble enzymes. Since most modification reactions are slow while their reverse reactions are rapid, modified tubulin is only detected on long-lived stable microtubules. Most of these modifications occur on the C014 Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Image of a fibroblast cell. Containing fluorescently labeled actin, red, and microtubules. Green. Terminal region of alpha-tubulin. This region, which is rich in negatively charged glutamate, forms relatively unstructured tails that project out from the microtubule and form contacts with motors. Thus, it is believed that tubulin modifications regulate the interaction of motors with the microtubule. Since these stable modified microtubules are typically oriented towards the site of cell polarity in interphase cells, this subset of modified microtubules provide a specialized route that helps deliver vesicles to these polarized zones. These modifications include deuterosination, the removal of the C-terminal TO sign from alpha-tubulin. This reaction exposes a glutamate at the new C-terminus. As a result, microtubules that accumulate this modification are often referred to as glue microtubules. Although the tubulin carboxypeptidase has yet to be identified, the tubulin tyrosine ligase (TTL) is known. 40. Delta 2, the removal of the last two residues from the C terminus of alpha-tubulin. 41. Unlike deuterosination, this reaction is thought to be irreversible and has only been documented in neurons. Acetylation, the addition of an acetyl group to lysine 40 of alpha-tubulin. This modification occurs on a lysine that is accessible only from the inside of the microtubule, and it remains unclear how enzymes access the lysine residue. The nature of the tubulin acetyltransferase remains controversial, but it has been found that in mammals the major acetyltransferase is AT1. 42. However, the reverse reaction is known to be catalyzed by DAC6. 43. The real impact of acetylation in the structure and function of microtubules remains elusive. 44. Polyglutamylation, the addition of a glutamate polymer, typically 4 to 6 residues long, 45, to the gamma-carboxyl group of any one of 5 glutamates found near the end of alpha-tubulin. Enzymes related to TTL add the initial branching glutamate, TTL4, 5 and 7, while other enzymes that belong to the same family lengthen the polyglutamate chain, TTL6, 11 and 13. 39. Polyglycylation, the addition of a glycine polymer, 2 to 10 residues long, to the gamma-carboxyl group of any one of 5 glutamates found near the end of beta-tubulin. TTL3 and 8 add the initial branching glycine, while TTL10 lengthens the polyglycine chain. 39. Tubulin is also known to be phosphorylated, ubiquitinated, sumoylated, and palmitoylated. 38. Tubulin binding drugs and chemical effects, edit. A wide variety of drugs are able to bind to tubulin and modify its assembly properties. These drugs can have an effect at intracellular concentrations much lower than that of tubulin. This interference with microtubule dynamics can have the effect of stopping a cell's cell cycle and can lead to. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Programmed cell death or apoptosis. However, there are data to suggest that interference of microtubule dynamics is insufficient to block the cells undergoing mitosis. 46. These studies have demonstrated that suppression of dynamics occurs at concentrations lower than those needed to block mitosis. Suppression of microtubule dynamics by tubulin mutations or by drug treatment have been shown to inhibit cell migration. 47. Both microtubule stabilizers and destabilizers can suppress microtubule dynamics. The drugs that can alter microtubule dynamics include the cancer-fighting taxane class of drugs, paclitaxel, Taxol, and docetaxel, block dynamic instability by stabilizing GDP-bound tubulin in the microtubule. Thus, even when hydrolysis of GTP reaches the tip of the microtubule, there is no depolymerization and the microtubule does not shrink back. Taxanes, alone or in combination with platinum derivatives, carboplatin, or gemcitabine, are used against breast and gynecological malignancies, squamous cell carcinomas, head and neck cancers, some lung cancers, etc. The epothelones, for example ixabepilone, work in a similar way to the taxanes. Vinorelbine, nocodazole, vincristine, and colchicine have the opposite effect, blocking the polymerization of tubulin into microtubules. Eribulin binds to the plus growing end of the microtubules. Eribulin exerts its anticancer effects by triggering apoptosis of cancer cells following prolonged and irreversible mitotic blockade. Expression of 3-tubulin has been reported to alter cellular responses to drug-induced suppression of microtubule dynamics. In general, the dynamics are normally suppressed by low, subtoxic concentrations of microtubule drugs that also inhibit cell migration. However, incorporating 3-tubulin into microtubules increases the concentration of drug that is needed to suppress dynamics and inhibit cell migration. Thus, tumors that express 3-tubulin are not only resistant to the cytotoxic effects of microtubule-targeted drugs, but also to their ability to suppress tumor metastasis. 48. Moreover, expression of 3-tubulin also counteracts the ability of these drugs to inhibit angiogenesis, which is normally another important facet of their action. 49. Microtubule polymers are extremely sensitive to various environmental effects. Very low levels of free calcium can destabilize microtubules, and this prevented early researchers from studying the polymer in vitro. 12. Cold temperatures also cause rapid depolymerization of microtubules. In contrast, heavy water promotes microtubule polymer stability. 50. Proteins that interact with microtubules, edit. Microtubule-associated proteins, MAPs, edit. Main article, Microtubule-associated protein. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. MAPs have been shown to play a crucial role in the regulation of microtubule dynamics in vivo. The rates of microtubule polymerization, depolymerization, and catastrophe vary depending on which microtubule-associated proteins, MAPs, are present. The originally identified MAPs from brain tissue can be classified into 2 groups based on their molecular weight. This first class comprises MAPs with a molecular weight below 55 to 62 kDa, and are called TO proteins. In vitro, TO proteins have been shown to directly bind microtubules, promote nucleation and prevent disassembly, and to induce the formation of parallel arrays. 51. Additionally, TO proteins have also been shown to stabilize microtubules in axons and have been implicated in Alzheimer's disease. 52. The second class is composed of maps with a molecular weight of 200 to 1000 K, of which there are 4 known types: map 1, map 2, map 3 and map 4. Map 1 proteins consists of a set of 3 different proteins, A, B and C. The C protein plays an important role in the retrograde transport of vesicles and is also known as cytoplasmic dynein. MAP2 proteins are located in the dendrites and in the body of neurons, where they bind with other cytoskeletal filaments. The MAP4 proteins are found in the majority of cells and stabilize microtubules. In addition to maps that have a stabilizing effect on microtubule structure, other maps can have a destabilizing effect either by cleaving or by inducing depolymerization of microtubules. 3 proteins called catenin, spastin, and phygetin have been observed to regulate the number and length of microtubules via their destabilizing activities. Furthermore, cracked-like protein is predicted to be localized to the microtubules. 53. Maps are determinants of different cytoskeletal forms of axons and dendrites, with microtubules being farther apart in the dendrites. 54. Plus N tracking proteins, plus tips, edit. Main article, microtubule plus N tracking protein. Plus N tracking proteins are map proteins which bind to the tips of growing microtubules and play an important role in regulating microtubule dynamics. For example, plus tips have been observed to participate in the interactions of microtubules with chromosomes during mitosis. The first map to be identified as a N tip was CLIP170, cytoplasmic linker protein, which has been shown to play a role in microtubule depolymerization rescue events. Additional examples of N tips include EB1, EB2, EB3, P150 glued, dynamitin, LES1, CLIP115, CLASP1, and CLASP2. Citation needed. Motor proteins, edit. Microtubules can act as substrates for motor proteins that are involved in important cellular functions such as vesicle trafficking and cell division. Unlike other microtubule-associated proteins, motor proteins utilize the energy from ATP hydrolysis to generate mechanical work that moves the protein along the substrate. The major motor proteins that interact with microtubules are kinsin, which usually moves toward the plus end of the microtubule, and dynein, which moves toward the end. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. A cytoplasmic dynein motor bound to a microtubule. A kinsin molecule bound to a microtubule. A 3D diagram of a centriole. Each circle represents one microtubule. In total there are 27 microtubules organized into 9 bundles of 3. Dynein is composed of 2 identical heavy chains, which make up 2 large globular head domains, and a variable number of intermediate and light chains. Dynein-mediated transport takes place from the plus end towards the end of the microtubule. ATP hydrolysis occurs in the globular head domains, which share similarities with the AAA+, AT-PASE. Associated with various cellular activities, protein family. ATP hydrolysis in these domains is coupled to movement along the microtubule via the microtubule binding domains. Dynein transports vesicles and organelles throughout the cytoplasm. In order to do this, dynein molecules bind organelle membranes via a protein complex that contains a number of elements including dynactin. Kinsin has a similar structure to dynein. Kinsin is involved in the transport of a variety of intracellular cargoes, including vesicles, organelles, protein complexes, and MNAs toward the microtubules, plus, end. 55. Some viruses, including retroviruses, herpesviruses, parvoviruses, and adenoviruses, that require access to the nucleus to replicate their genomes attached to motor proteins. Mitosis, edit. Centrosomes, edit. The centrosome is the main mitoc, microtubule, organizing center, of the cell during mitosis. Each centrosome is made up of two cylinders called centrioles, oriented at right angles to each other. The centriole is formed from 9 main microtubules, each having 2 partial microtubules attached to it. Each centriole is approximately 400 nm long and around 200 nm in circumference. 56. The centrosome is critical to mitosis as most microtubules involved in the process originate from the centrosome. The minus ends of each microtubule begin at the centrosome, while the plus explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. This diagram depicts the organization of a typical mitotic spindle found in animal cells. Shown here are the 3 main types of microtubules during mitosis and how they are oriented in the cell and the mitotic spindle. Ends radiate out in all directions. Thus the centrosome is also important in maintaining the polarity of microtubules during mitosis. 57. Most cells only have one centrosome for most of their cell cycle. However, right before mitosis, the centrosome duplicates, and the cell contains 2 centrosomes. 58. Some of the microtubules that radiate from the centrosome grow directly away from the sister centrosome. These microtubules are called astral microtubules. With the help of these astral microtubules, the centrosomes move away from each other towards opposite sides of the cell. Once there, other types of microtubules necessary for mitosis, including interpolar microtubules and K-fibers, can begin to form. 59. A final important note about the centrosomes and microtubules during mitosis is that while the centrosome is the mitoc for the microtubules necessary for mitosis, research has shown that once the microtubules themselves are formed and in the correct place, the centrosomes themselves are not needed for mitosis to occur. 60. Microtubule subclasses, edit. Astral microtubules are a subclass of microtubules which only exist during and around mitosis. They originate from the centrosome, but do not interact with the chromosomes, kinetochores, or with the microtubules originating from the other centrosome. 61. Instead, their microtubules radiate towards the cell membrane. Once there, they interact with specific motor proteins which create force that pull the microtubules, and thus the entire centrosome, towards the cell membrane. As stated above, this helps the centrosomes orient themselves away from each other in the cell. However, these astral microtubules do not interact with the mitotic spindle itself. Experiments have shown that without these astral microtubules, the mitotic spindle can form. However, its orientation in the cell is not always correct, and thus mitosis does not occur as effectively. 62. Another key function of the astral microtubules is to aid in cytokinesis. Astral microtubules interact with motor proteins at the cell membrane to pull the spindle and the entire cell apart once the chromosomes have been replicated. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Interpolar/polar microtubules are a class of microtubules which also radiate out from the centrosome during mitosis. These microtubules radiate towards the mitotic spindle, unlike astral microtubules. Interpolar microtubules are both the most abundant and dynamic subclass of microtubules during mitosis. Around 95% of microtubules in the mitotic spindle can be characterized as interpolar. Furthermore, the half-life of these microtubules is extremely short, as it is less than 1 minute. 63. Interpolar microtubules that do not attach to the kinetochores can aid in chromosome congregation through lateral interaction with the kinetochores. 64. K-fibers /kinetochore microtubules are the third important subclass of mitotic microtubules. These microtubules form direct connections with the kinetochores in the mitotic spindle. Each K-fiber is composed of 20-40 parallel microtubules, forming a strong tube which is attached at one end to the centrosome and on the other to the kinetochore, located in the center of each chromosome. Since each centrosome has a K-fiber connecting to each pair of chromosomes, the chromosomes become tethered in the middle of the mitotic spindle by the K-fibers. K-fibers have a much longer half-life than interpolar microtubules, at between 4 and 8 minutes. 65. During the end of mitosis, the microtubules forming each K-fiber begin to dissociate, thus shorting the K-fibers. As the K-fibers shorten, the pair chromosomes are pulled apart right before cytokinesis. Previously, some researchers believed that K-fibers form at their minus end originating from the centrosome. Just like other microtubules, however, new research has pointed to a different mechanism. In this new mechanism, the K-fibers are initially stabilized at their plus end by the kinetochores and grow out from there. The minus end of these K-fibers eventually connect to an existing interpolar microtubule and are eventually connected to the centrosome in this way. 66. Microtubule nuclear in the mitotic spindle, edit. Most of the microtubules that form the mitotic spindle originate from the centrosome. Originally it was thought that all of these microtubules originated from the centrosome via a method called search and capture, described in more detail in a section above. However, new research has shown that there are additional means of microtubule nucleation during mitosis. One of the most important of these additional means of microtubule nucleation is the RUN-GTP pathway. RUN-GTP associates with chromatin during mitosis to create a gradient that allows for local nucleation of microtubules near the chromosomes. Furthermore, a second pathway known as the augmin/Hors complex. Some organisms use the more studied augmin complex, while others such as humans use an analogous complex called Hors. acts an additional means of microtubule nucleation in the mitotic spindle. 66. Functions, edit. Cell migration, edit. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Microtubule plus ends are often localized to particular structures. In polarized interface cells, microtubules are disproportionately oriented from them toward the site of polarity, such as the leading edge of migrating fibroblasts. This configuration is thought to help deliver microtubule bound vesicles from the Golgi to the site of polarity. Dynamic instability of microtubules is also required for the migration of most mammalian cells that crawl. 67. Dynamic microtubules regulate the levels of key G proteins such as ROA, 68, and RAC1, 69, which regulate cell contractility and cell spreading. Dynamic microtubules are also required to trigger focal adhesion disassembly, which is necessary for migration. 70. It has been found that microtubules act as struts that counteract the contractile forces that are needed for trailing edge retraction during cell movement. When microtubules in the trailing edge of cell are dynamic, they are able to remodel to allow retraction. When dynamics are suppressed, microtubules cannot remodel and, therefore, oppose the contractile forces. 47. The morphology of cells with suppressed microtubule dynamics indicate that cells can extend the front edge, polarized in the direction of movement, but have difficulty retracting their trailing edge. 71. On the other hand, high drug concentrations, or microtubule mutations that depolymerize the microtubules, can restore cell migration, but there is a loss of directionality. It can be concluded that microtubules act both to restrain cell movement and to establish directionality. Cilia and flagella, edit. Microtubules have a major structural role in eukaryotic cilia and flagella. Cilia and flagella always extend directly from mTOC, in this case termed the basal body. The action of the dynein motor proteins on the various microtubule strands that run along a cilium or flagellum allows the organelle to bend and generate force for swimming, moving extracellular material, and other roles. Prokaryotes possess tubulin-like proteins including FTSZ. However, prokaryotic flagella are entirely different in structure from eukaryotic flagella and do not contain microtubule-based structures. Development, edit. The cytoskeleton formed by microtubules is essential to the morphogenetic process of an organism's development. For example, a network of polarized microtubules is required within the oocyte of Drosophila melanogaster during its embryogenesis in order to establish the axis of the egg. Signals sent between the follicular cells and the oocyte, such as factors similar to epidermal growth factor, cause the reorganization of the microtubules so that their ends are located in the lower part of the oocyte, polarizing the structure and leading to the appearance of an anterior posterior axis. 72. This involvement in the body's architecture is also seen in mammals. 73. Another area where microtubules are essential is the development of the nervous system in higher vertebrates, where tubulin's dynamics and those of the associated proteins, such as the Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Microtubule-associated proteins are finely controlled during the development of the nervous System. 74. Gene regulation, edit. The cellular cytoskeleton is a dynamic system that functions on many different levels, in addition to giving the cell a particular form and supporting the transport of vesicles and organelles. It can also influence gene expression. The signal transduction mechanisms involved in this communication are little understood. However, the relationship between the drug-mediated depolymerization of microtubules and the specific expression of transcription factors has been described, which has provided information on the differential expression of the genes depending on the presence of these factors. 75. This communication between the cytoskeleton and the regulation of the cellular response is also related to the action of growth factors. For example, this relation exists for connective tissue growth factor. 76. See also, edit. Microtentacle. Orchestrated objective reduction, a hypothesis explaining consciousness. References, edit. 1 digital downloads. Per Solutions. Archived from the original on the 29th of September 2022. Retrieved the 20th of February 2020. 2 Ledbetter MC, Porter Kr. 1963. A microtubule in plant cell fine structure. Journal of Cell Biology. 19, 123952. 10.1083/JCB.19.1.239. PMC 2106853. PMID 1986665. 3 Chalf M. Thomson J.N. 1979. Organization of neuronal microtubules in the nematode. Caenorhabditis elegans. Journal of Cell Biology. 82, 127889 do. 10.1083/JCB.82.1.278. PMC 2110421. PMID 479300. 4 Diwan JJ, 2006. Microtubules. Rensselaer Polytechnic Institute. Archived from the original on the 6th of February 2014. Retrieved the 24th of February 2014. 5 Vale Road, February 2003. The Molecular Motor Toolbox for Intracellular Transport. Cell 112, 4. 46780. Do. 10.1016/S0092-8674, 03-00111-9. PMID 126003111. S2 SID 151000327. 6 Howard J. Hyman A. February 2007. Microtubule polymerases and depolymerases. Current Opinion in Cell Biology. 19, 13152, 10.1016/J.Seb.2006.12.009. PMID 1718496. 7 Gian Guess, Narita, Pop D, Ghosh Dastida U, Li LJ, Srinivasan R, Balaju Brahmanian MK, Adati, Co. F, Larson M, Robinson RC, March 2016. Novel actin filaments from Bacillus thuringiensis form nanotubules for plasmid DNA segregation. Proceedings of the National Academy of Sciences of the United States of America. 113, 9E 1200-5. Bibcode 2016 NAS 113E1200J. Do 10.1073/NAS.1600129113. PMC 4780641. PMID 268731105. 8 Wayne, R. 2009. Plant Cell Biology, From Astronomy to Zoology, archived 21 February 2024 at the Wayback Machine. Amsterdam, Elsevier/Academic Press, p. 165. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 9 Cooper GM, 2000. Microtubule Motors and Movements. The Cell, A Molecular Approach. 2nd edition. Archived from the original on 3 December 2021. Retrieved 12 March 2019. 10 Kapil v. Hurst W.G., Henschel C., Prebis Chess, Reber S., March 2019. M-Track, Automated Detection, Tracking, and Analysis of Dynamic Microtubules. Scientific Reports. 9, 13794. Bibcode 2019 NAT SR 9.3794K. Do 10.1033/S41598-018-37767-1. PMC 6405942. PMID 308467005. 11 Low J, Lee H., Downing K.H., No Gale Z., November 2001. Refined structure of alpha-beta tubulin at 3.5 A resolution. Journal of Molecular Biology. 313, 5 1045 57 do. 10.1006/B.2001.5077. PMID 1170061. Archived from the original on 22 January 2021. Retrieved 9 September 2019. 12 ABC Weisenberg R.C., September 1972. Microtubule formation in vitro in solutions containing low calcium concentrations. Science. 177, 4054 1104 5. Bibcode 1972 SCI 177.1104W. Do 10.1126/science.177.4054.1104. PMID 46266639. S2 SID 34875893. 13 Desio, Mitchison T.J., 1997. Microtubule Polymerization Dynamics. Annual Review of Cell and Developmental Biology. 13, 83117. Do 10.1046/anav.cellbio.13.1.83. PMID 9442869. 14 Desai, A. Mitchison, T.J. 1997. Microtubule Polymerization Dynamics. Annual Review of Cell and Developmental Biology. 13, 83117. Do 10.1046/anav.cellbio.13.1.83. ISSN 1081-0706. PMID 9442869. 15 Caban, Bruhard GJ, 2017. A microtubule bestiary, structural diversity in tubulin polymers. Molecular biology of the cell. 28, 22 2924 31 do. 10.1091/mbc.e16-05-0271. PMC 5662251. PMID 290849110. 16 Cretian D, Mitars F, Verdi F, Carcenti E, Wade R.H., June, 1992. Lattice defects in microtubules. Protofilament numbers vary within individual microtubules. Journal of Cell Biology. 117, 5103140. Do. 10.1083/JCB.117.5.1031. PMC 2289483. PMID 1577866. 17 Walkera, Abrian E.T., Pryor N.K., Sabero M.F., Vota W.A., Erickson H.P., Simon ed., October, 1988. Dynamic instability of individual microtubules analyzed by video light microscopy, rate constants and transition frequencies. The Journal of Cell Biology. 107, for 1437-48. Cite CX 10.1.1.525.507. Do. 10.1083/JCB.107.4.1437. PMC 2115242. PMID 3170635. 18 Sway H, Downing K.H., August, 2010. Structural basis of interprotofilament interaction and lateral deformation of microtubules. Structure. 18, 8 1022 31 do. 10.1016/J. Street 2010.05.010. PMC 2976607. PMID 206964002. 19 Basson D.M., Hu Y., Bowser S.S., Benar Valley N.K., August 2016. Maintenance of electrostatic stabilization in altered tubulin lateral contacts may facilitate formation of helical filaments in foraminifera. Scientific reports. 6, 31723. Bibcode, 2016 Nat SR, 631723B. Do. 10.1038/SREP31723. PMC 4990898. PMID 275393992. 20 Nogale Z, 2000. Structural Insights into Microtubule Function. Annual Review of Biochemistry. 69. 277302. Do. 10.1011/anav.biochem.69.1.277. PMID 1096640. 21 Schlepper D, Oliver Ma, Andrew J. M., Low J., June, 2005. Structure of bacterial tubulin BDUB/A/B. Evidence for horizontal gene transfer. Proceedings of the National Academy of Sciences of the United States of America. 102, 26 9175. Bibcode, 2005 NAS 102.9170s. Do. 10.1073/NAS.05028599102. PMC 11666614. PMID 159679998. 22 Pilhofer M, Ladinsky MS, McDowell AW, Petroni G, Jensen GJ, December 2011. Microtubules in bacteria. Ancient tubulins build a 5-protofilament homologue of the eukaryotic cytoskeleton. PLOS Biology 9, 12E10012113 do 10.1371/journal.bio.10012113. PMC 3232192. PMID 22162949. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 23 Medical definition of neurotubules. www.merriam-webster.com. Archived from the original on the 27th of September 2018. Retrieved the 26th of September 2018. 24 Joule B, Maker DP, Scarenberg R, Koenig T, Schwank B, Kobler O, Windhorst S, Crews Mr. Mikhailover M, Calderon de Ander F, August 2017. Microtubules modulate F-actin dynamics during neuronal polarization. Scientific reports 7, 19583. Bibcode 2017 NAT SR 7.9583Z. Do 10.1038/S41598-017-09832-8. PMC 557506-2. PMID 2885198-2. 25 Bartolini F, Gunderson GG, October 2006. Generation of non-centrosomal microtubule arrays. Journal of Cell Science. 119, pt 24155-63 do. 10.1242/JCS.03227. PMID 1703854-2. 26 Desire, Mitchison TJ, 1997. Microtubule Polymerization Dynamics. Annual Review of Cell and Developmental Biology. 13, 83117. Do 10.1046/anav.cellbio.13.1.83. PMID 9442869. 27 Vinogradova T, Miller PM, Kavrina I, July 2009. Microtubule Network Asymmetry in Motile Cells, Role of Golgi-derived Array. Cell Cycle 8, 14 2168-74 do. 10.4161/cc.8.14.9074. PMC 3163838. PMID 195568595. 28 Yahara R, Nozawa RS, Tomaoka Petri S, Vale Road, Obus C, Goshima G, April 2009. The Augmin complex plays a critical role in spindle microtubule generation for mitotic progression and cytokinesis in human cells. Proceedings of the National Academy of Sciences of the United States of America. 106, 17 6998 7003. Bibcode 2009 as 106.6998 U. Do 10.1073/nas.09015871106. PMC 2668966. PMID 193691998. 29 Alberts B, Johnson O, Lewis J, Raff M, Roberts K, Walter P, 2002. The Self-Assembly and Dynamic Structure of Cytoskeletal Filaments. Molecular Biology of the Cell, 4th ed. New York, Garland. Science. Archived from the original on 5 June, 2018. Retrieved 5 September, 2017. 30 Carp G, 2005. Cell and Molecular Biology, Concepts and Experiments. USA, John Wiley and Sons. p. 355. ISBN 978-0-471-46580-5. 31 Weisenberg R.C., Deary W.J., Dickinson P.J., September 1976. Tubulin nucleotide interactions during the polymerization and depolymerization of microtubules. Biochemistry. 15, 19 4248 54. Do 10.1021/by00664018. PMID 963034. 32 Mitchison T, Kirschner M, 1984. Dynamic Instability of Microtubule Growth. Nature. 312, 5991. 23742. Bibcode, 1984 Nature.312237M. Do 10.1038/312237A0. PMID 6504138. S2 SID 30079133. 33 Kirschner M, Mitchison T, May 1986. Beyond Self-Assembly, From Microtubules to Morphogenesis. Cell. 45, 332942 Do. 10.1016/0092-8674, 86, 90318-1. PMID 35164113. S2 SID 36994346. 34 Cheeseman M, Daysire, January 2008. Molecular Architecture of the Kinetica Microtubule. Interface. Nature Reviews. Molecular Cell Biology. 9, 13346 Do. 10.1038/NRM 2310. PMID 180974444. S2 SID 34121605. 35 AB Infante as, Stein MS, Chai Y, Borisi GG, Gunderson GG, November 2000. Detyrogenated glue. Microtubules are stabilized by an ATP-sensitive plus end cap. Journal of Cell Science. 113, 22. 3907-19. Do. 10.1042/JCS.113.22.3907. PMID 11058078. Archived from the original on 2024-02. 21 retrieved 23 June, 2014. 36 Palazzo F, Kukhtar, Alberts as, Gunderson GG, August 2001. MDA mediates Rho-regulated formation and orientation of stable microtubules. Nature Cell Biology 3.87239. Do 10.1038/3508735. PMID 11483957. S2 SID 7374170. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 37 Wen Y, Eng Ch, Shmranza J, Cabrera Poc N, Morris E J, Chen M, Wallaby J, Alberts Az. Gunderson G G, September 2004. EB1 and APC bind to MDA to stabilize microtubules downstream of Rho and promote cell migration. Nature Cell Biology 6.982030 Do 10.1038 /NCB1160. PMID 15311282. S2 SID 292141110. 38 AB Jank C, Bulinski JC, November 2011. Post-translational regulation of the microtubule. Cytoskeleton, mechanisms and functions. Nature Reviews. Molecular Cell Biology 12.1777386. Do 10.1038 /NRM 3227. PMID 22086369. S2 SID 5969290. 39 ABC Garnham CP, Roll Mekako, July 2012. The chemical complexity of cellular microtubules. Tubulin post-translational modification enzymes and their roles in tuning microtubule functions. Cytoskeleton 69, 744263 Do 10.1002 /cm.21027. PMC 3459347. PMID 224222711. 40 Usfeld K, Wayland J, Plusman U, Dodermunt H, Jerk V, Weber K, February 1993. Characterization of the tubulin tyrosine ligase. The Journal of Cell Biology. 120, 372532. Do 10.1083 /JCB.120.3.725. PMC 2119537. PMID 8093886. 41 Pato Lafaniche L, Eddie B, Dinule P, Van Dorsaleo, Mazaguil H, Lakaya J P, Wayland J. Job D, October 1991. Characterization of a major brain tubulin variant which cannot be tyrosinated. Biochemistry 30, 43105238 Do 10.1021 /by001070022. PMID 19319774. 42 Kalabic N, Sorrentino S, Pelas E, Bolasco G, Martin S. C, Heppenstall Pa, 10 June 2013. TAT1 is the major tubulin acetyltransferase in mice. Nature Communications 4, 1962. Bibcode 2013 Natco 4.1962K. Do 10.1038 /MCOMS 2962. PMID 237489001. 43 Hubbard C, Guardiola O, Shao R, Kawagachi Y, Ito O, Nixon O, Yoshida M, Wang XF, Yao TP, May 2002. DAC6 is a microtubule-associated destylase. Nature 417, 6887 4558. Bibcode 2002 nature.417455H. Do 10.1038/417455A. PMID 12024216. S2 SID 4373254. 44 Carmina, Bruno, Marinho, H. Susanna, Matus, Katarina Lopes, Nolasco, Sophia, Soares, Helena. 2023. Tubulin post-translational modifications, the elusive roles of acetylation. Biology 12, 4. 561 do 10.3390/biology 12040561. ISSN 2079-7737. PMC 101360995. PMID 37106761. 45 Albert S., Dispriyez, Gruzchinsky C., Kalakofer, Gross F., Denule P., Edi B., June 1993. Reversible polyglutamylation of alpha and beta tubulin and microtubule dynamics in mouse brain. Neurons. Molecular biology of the cell. 4, 661526 do 10.1091/MBC.4.6.615. PMC 300968. PMID 81040553. 46 Ganglia, Yang H., Cabral F., November 2010. Paclitaxel-dependent cell lines reveal a novel drug. Activity. Molecular cancer therapeutics. 9, 11 2914 23 do 10.1158/7135-7163. MCT 10-0552. PMC 2978777. PMID 209786163. 47 AB Yang H., Ganglia, Cabral F., October 2010. Inhibition of cell migration and cell division. Correlates with distinct effects of microtubule inhibiting drugs. The Journal of Biological Chemistry. 285, 42 3224250 do 10.1074/JBC.M110.160820. PMC 2952225. PMID 2069677. 48 Altanzi, Mohammed, Ganguly, Ainutash, Amrain, Mavias, Simonowicz, Philip, Lee, Shu, Lawson. Gills, March 2020. Beta-3 tubulin is critical for microtubule dynamics, cell cycle regulation, and spontaneous release of microvesicles in human malignant melanoma cells, a 375. International Journal of Molecular Sciences. 21, 1656 do 10.3390/IGMS 210516656. PMC 7084453. PMID 3212195. 49 Ganguly, Ainutash, Yang, Hailing, Fernando, Gabriel, May 2011. Class 3 tubulin counteracts the ability of paclitaxel to inhibit cell migration. Oncotarget 2, 5368377. Do 10.18632/oncotarget.250. PMC 3248193. PMID 215767662. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 50 Burgess J., Northcote DH, September 1969. Action of colchicine and heavy water on the polymerization of microtubules in wheat root meristem. Journal of Cell Science. 5. 243351. Do. 10.1242/JCS.5.2.433. PMID 5362335. 51 Mandelkow E., Mandelkow M., February 1995. Microtubules and microtubule-associated proteins. Current opinion in cell biology. 7. 17281 Do. 10.1016/0955-0674, 95, 80047-6. PMID 7755992. 52 Bramblett GT, Goedet M., Jakes R., Merrick SE, Trojanowski JQ, Lee VM, June 1993. Abnormal toe phosphorylation at SA396 in Alzheimer's disease recapitulates development and contributes to reduced microtubule binding. Neuron 10.6.1089-99 Do. 10.1016/0896-6273, 93, 90057X. PMID 8318230. S2 SID 231808847. 53 The Human Protein Atlas. www.proteinatlas.org. Archived from the original on 1 May 2017. Retrieved 27 April 2017. 54 Hirakawa, N. 1994. The neuronal cytoskeleton, roles in neuronal morphogenesis and organelle transport. Molecular neurobiology, mechanisms common to brain, skin and immune system. Series. Progress in clinical and biological research. Willis, Inc. 390, 117143. PMID 7536943. 55 Hirakawa N., Noda Y., Tanaka Y., Niwa S., October 2009. Kinson superfamily motor proteins and intracellular transport. Nature Reviews. Molecular cell biology. 10.10.68296. Do. 10.1038/NRM 2774. PMID 1977370. S2 SID 181299292. 56 Marshall W.F., Rosenbaum J.L., March 1999. Cell Division, The Renaissance of the Centriole. Current Biology. 9, 6R21820. Bibcode, 1999 CBio, 9.R218M. Do. 10.1016/S0960-9822, 99, 80133. X. PMID 1020987. S2 SID 169512668. 57 Per G, Schibeli, February 1997. Centrosome microtubule nucleation. Journal of Cell Science. 110, PT 3295300 Do. 10.1242/JCS.110.3.295. PMID 9057082. 58 Hinchcliffe A., Sluder G., May 2001. It Takes Two to Tango, Understanding How Centrosome Duplication is Regulated Throughout the Cell Cycle. Genes and Development. 15, 10.116781. Do. 10.1101/GAD.8940001. PMID 11358861. 59 4th S., Kapoor M., June 2017. The Mechanics of Microtubule Networks in Cell Division. The Journal of Cell Biology. 216, 6 1525 1531 do. 10.1083/JCB.2016122064. PMC 5461028. PMID 2849044. 60 Khojakov, A., Cole, R.W., Oakley, B.R. and Reader, C.L. 2000. Centrosome independent mitotic spindle formation in vertebrates. Car. Biol. 10, 5967. Do 10.1016/S0960-9822, 99, 00276-6. 61 Rosenblatt J., March 2005. Spindle assembly, asters part their separate ways. Nature Cell Biology. 7, 321922 do. 10.1038/NCB0305-219. PMID 1573894. S2 SID 8082479. 62 Noblik J.A., December 2010. Asymmetric Cell Division, Recent Developments and Their Implications. For Tumor Biology. Nature Reviews. Molecular Cell Biology. 11, 12.84960. Do. 10.1038/NRM3010. PMC 3941022. PMID 2110261. 63 J.Y., Kronbusch P.J., Borisy G.G., November 1995. Kinetica of Microtubule Dynamics and the Metaphase-anaphase Transition. The Journal of Cell Biology. 131, 372134. Do. 10.1083/JCB.131.3.721. PMC 2120628. PMID 7593192. 64 CAIS, O'Connell C.B., Khojakov, Walchak C.E., July 2009. Chromosome congression in the absence of kinetica fibers. Nature Cell Biology. 11, 78328 do. 10.1038/NCB1890. PMC 2895821. PMID 195253938. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 65 Backhaum SF, Thomson S.L., Manning L., Compton the, January 2009. Genome stability is ensured by temporal control of kinetica microtubule dynamics. Nature Cell Biology. 11, 12735. Do. 10.1038/NCB1809. PMC 2614462. PMID 19060894. 66 AB Mania S. Vernos I. June 2012. Microtubule assembly during mitosis, from distinct origins to distinct functions. Journal of Cell Science. 125, pt 12 2805 14 do, 10.1242/JCS.0924229. PMID 227360044. 67 Mikhailov, Gunderson G.G., 1998. Relationship between microtubule dynamics and lamellipodium. Formation revealed by direct imaging of microtubules in cells treated with no codazole or taxol. Cell motility and the cytoskeleton. 41, 4325 40 do, 10.1002 CISI, 1097-0169, 1998, 41:43.0.co, 2d. PMID 9858157. 68 Ren XD, Keosis WB, Schwartzma, February 1999. Regulation of the small GTP binding protein. Row by cell adhesion and the cytoskeleton. The EMBO Journal. 18, 357885. Do. 10.1093/embodge/18.3.578. PMC 1171150. PMID 9927417. 69 Waterman Store centimeter, Worthilake Ra, Lou BP, Burridge K, Salmon ed., May 1999. Microtubule. Growth activates RAC1 to promote lamellipodile protrusion in fibroblasts. Nature Cell Biology. 1, 145. 50 do, 10.1038/9018. PMID 105598663. S2 SID 263211103. 70 Esrati EJ, Partridge Ma, Gunderson GG, June 2005. Microtubule-induced focal adhesion. Disassembly is mediated by dynamin and focal adhesion kinase. Nature Cell Biology. 7, 658-190. Do. 10.1038/NCB1262. PMID 158950076. S2 SID 37153935. 71 Ganglia, Yang H, Sharma R, Patel KD, Cabral F, December 2012. The role of microtubules and their dynamics in cell migration. The Journal of Biological Chemistry. 287, 52433569. Do. 10.1074/JBC.M112.423905. PMC 3527923. PMID 2313528. 72 Van Eden F, St. Johnston D, August 1999. The polarization of the anterior, posterior and dorsal ventral axes during Drosophila. Current opinion in genetics and development. 9, 4396. 404 do. 10.1016/S0959-437X, 99, 80060-4. PMID 104495356. 73 Beddington RS, Robertson E.J., January 1999. Axis development and early asymmetry in mammals. Cell 96, 2195209 do. 10.1016/S0092-8674, 00-80560-7. PMID 9988215. S2 SID 16264083. 74 Tucker RP, 1990. The roles of microtubule-associated proteins in brain morphogenesis, a review. Brain Research. Brain Research Reviews. 15, 210120 do. 10.1016/0165-0173, 90, 90013E. PMID 2282447. S2 SID 12641708. 75 Rosette C., Karen M., March 1995. Cytoskeletal control of gene expression, depolymerization of microtubules activates NF-kappa B. The Journal of Cell Biology. 128, 61111 do. 10.1083/JCB.128.6.1111. PMC 2120413. PMID 7896875. 76 OTTC, I1CD, Grainus O, Geil K, Goppeltstrube M, November 2003. Modulation of the expression of connective tissue growth factor by alterations of the cytoskeleton. The Journal of Biological Chemistry. 278, 4544300511 do. 10.1074/JBC.M3091402000. PMID 12951326. External links, edit. MB Info, Microtubules. 3D microtubule structures in the M Data Bank, EMDB. Protocols for generating microtubules. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Categories: Cell anatomy, cytoskeleton. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.